Hello, everyone, and welcome to today's webinar. And thank you to everybody who has joined us today. This is the very first webinar of Obesity 2020, a joint webinar series brought to you by Inside Scientific and the American Physiological Society. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. Between now and December, we have a number of webinars lined up, all focused on the science being conducted by leading obesity researchers around the world. Today's webinar is titled Brain Circuits Driving Appetite and will feature Professor Laura Heisler from the Rowett Institute at the University of Aberdeen in Scotland. Professor Heisler will discuss how our brain circuits control body weight, for example, through the adipocyte hormone leptin. She will also review how our genes impact our waistline and how obesity medications capitalize on neurobiology to promote satiety, reduce hunger, and decrease body weight. So I'm sure everybody's looking forward to getting started here, but first we'd like to acknowledge our partners at the APS and thank the sponsors for making it all possible. And so without any further ado, I'm very pleased to welcome Professor Laura Heisler. Laura, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Great. Well, thank you. And thank you so much for the invitation to speak to all of you today about one of my favorite topics, which is appetite. So hello from Scotland, and here we go. You can see here just on this starting slide, a little bit of ice cream, which I've been enjoying in these the beginning of the summer here. But why am I interested in appetite, and why is it important to study appetite? Well, that's because something that you may have noticed is that around the world, obesity is becoming more and more prevalent. And in fact, it's increased fourfold just over the past 20 years. And if you look at this world map of obesity and overweight, what you can see here is that the countries that are in the darker orange have higher rates of overweight and obesity, with the ones in the darkest orange being greater than 60% of people being overweight or obese. And this is a real concern because obesity predisposes to major medical illness. But the biggest concern is that obesity is increasing at the fastest rate in children. So the current rates of overweight and obesity, So I just mentioned that one of the problems is that obesity is increasing at the fastest rate in children. And what you might be really surprised to see is that in reception age children, so those are kids who are just starting school, so they're around 4 or 5 years old, already in those young children, we have rates of obesity and overweight as 1 in 4 of those children. And if you look when kids are then finishing primary school, so now kids are around 11 or 12, now 1 in 3 children are overweight or obese. And when you look at adults, now it's 2 to 3. And so what you can see is that obesity and overweight are becoming the new normal. And what I mean by that is that more people are overweight or obese than are a healthy weight. And this is going to have cost implications because obesity is associated with increasing major medical illness. So that's going to have a cost in not only to our hospitals, but also to our wider economy, because if people are ill, then they won't be going to work. So it's a personal cost in terms of health, but then it's also a financial cost to our hospitals and also to our economy. So what do we mean by overweight and obesity? How are these things defined? But there's something that's called body mass index, or BMI. And BMI is calculated by looking at a person's height, and based on that height, what's the expected healthy weight? And so when we look at this, you can look at your height and your weight, and you can see where you fall within this range. So, up to 18 is underweight. From 19 to 23 or 24 is a healthy weight. So 19 to 24 is a healthy weight. From 25 to 29 is overweight. 30 to 40 or 30 to 39 is obese, and 40 and above is extremely obese. And so what is probably, I think most people find it to be a little bit of a surprise where they fall within this scale, because looking around, you judge yourself, to what you see around you. And many people say, oh, I look okay. And then are quite surprised to find that they're in the, overweight category or lower obese category. And the reason why this matters, it absolutely doesn't matter how people look. The reason why this matters is because having excess body fat, having excess adiposity is bad for our health. We don't fully understand why it's bad for our health. But being overweight or obese predisposes to a variety of different diseases, such as type 2 diabetes, heart disease, cancer, gallbladder disease, etc. And this is why obesity is being thought of as one of the key global healthcare challenges of the 21st century. And I mentioned that obesity predisposes to cancer, and that's a bit of a surprise to many people. And in fact, it's such a surprise that Cancer Research UK has started with this ad campaign that you might have seen around the United Kingdom, trying to let people know that obesity is a risk factor for cancer. And in fact, lots of different types of cancer, 13 different types of cancer. With, in this schematic here, the larger circles are the ones where there's a stronger association with cancer and overweight and obesity. So this is why we need to be concerned about overweight and obesity. So how does it work? Well, the way that it works, the way that body weight is determined is a combination of what we take in, so our food intake, and our energy expenditure, so our energy in and our energy out. So how does this look? So you might hear in the media, you'll hear people talking about, well, the reason why we're becoming more obese or overweight is because people aren't exercising as much. Or you'll hear people say, well, the reason why we're becoming obese or overweight is because we're eating too much. But in fact, if you think about it as a equation or as a balance, eating more than what your body requires will cause weight gain, and eating less than your body requires will cause weight loss. If you reduce your physical activity because you now have a desk job, you'll need fewer calories. So you'll need to adjust the number of calories that you're taking in because your body doesn't require as many calories than if you were more active. So the way that diets work, all diets, it doesn't matter if it's the caveman diet or the GI diet or whatever type of diet, the way that all diets work is having fewer calories than the body requires to run. And so by taking in fewer calories than the body requires, the body then liberates energy from stores, and those stores are commonly in fat. And so by reducing body fat, you get a reduction in body weight. That's how you get your weight loss. So it doesn't really matter which diet you use. Any diet should work fine. It's basically whatever diet you can stick to, whichever one works best for you in your lifestyle. But more commonly the case is taking in more calories than we require, than our body requires. And you might imagine that you'd have to take in loads more calories than your body requires to get weight gain. But no, it doesn't need to be many. It can just be a few over time will lead to weight gain. So this next slide shows you just kind of this idea of this balance between taking energy in and energy expenditure. So if you've had all the calories that your body needs, and then you take in a little bit more, then, for example, if you had a Big Mac but you didn't really need a Big Mac, that would be equivalent to over 2 hours or 3 hours of walking. And if you just have a chocolate bar, for example, it takes me, 60 seconds to eat a chocolate bar, that's equivalent to 35 minutes of cycling. How about a yogurt? Oh, I just quite fancy a yogurt. Just want to eat something, just a little bit bored in front of my computer. That's the same as 40 minutes of swimming. A bag of chips, again, could be 45 minutes of gardening. So you get the idea that when we think about, it's very easy to take food in. It's very fast and it's very easy to take food in, but it's a lot harder to expend that energy with physical activity. So that's why many experts will say, if you're thinking about trying to change your body weight, that an easier way of tackling that is by adjusting your food intake, reducing your food intake. Of course, it's best to do a combination of both, but there's an expression: you can't outrun a bad diet. So, if you're having multiple additional Big Macs and mac and cheese and variety of different things, you'd have to be exercising 8-10 hours a day to try and make that equivalent. So it's trying to be aware of the calories that are in food, and most people don't have that full sort of awareness. So there are many websites that you can log into where it will tell you what is it equivalent to, what's this little treat that I want to have equivalent to with regards to running or cycling or something like that. So it can just being informed can help. When we think about obesity, though, we've got the diet, the food that I've just talked about. We've got exercise that I've just talked about, but we also have genes. And when we look to genes, especially like these monogenic syndromes or specific genetic mutations that people or animals may have, it gives us clues as to these key pathways that are regulating our appetite and our body weight. So I'm just going to show you a quick video which has an interview with a patient who has one of these monogenic syndromes.
50 years ago, never would have thought it was possible. The only way to view a brain was under a microscope after it had been removed from whoever or whatever. But now IT technology has pushed us to a point of understanding that we never thought, I think, we would ever get to. What's happened with the advent of imaging is that we're able to view the brain in action as it's actually carrying out processes. One of my areas of research is Prader-Willi syndrome. Probably what it's best known for is the overconsumption of food and the consequential obesity that comes with that. Society's stigmatized view of obesity generally means that people take the opinion that it's a consequence of moral frailty. Somebody is obese because they eat too much. But what we're discovering now with the advent of imaging is that there may be a reason why those people eat too much. If we take a regular individual and put them into the fMRI machine, and such an individual has just consumed a large meal and has reached that point of feeling full and sitting back, you'll find that although there is a neurological response to an image of a certain piece of food, it is by no means as great as it was when they were hungry. With Prader-Willi syndrome patients, that doesn't occur. They're going to be as equally responsive neurologically to an image of food when they're hungry as when they've just consumed a meal. It doesn't matter what they've just eaten or when they've just eaten, they're always going to be stimulated by food.
When I eat a meal, I feel full first of all, but then in 2 hours' time I feel as I haven't ate something. It feels frustrating being hungry all the time. If we put on too much weight, we can die.
The fMRI data really helps us pinpoint where the problem is. So now we have a target area and a target problem. The fullness signals aren't strong enough. We're hoping to overcome this with an implant that could artificially stimulate activity in that area. If we could do that, and we could release people from the need to have to think about food all the time and really help them lead more independent lives.
I think it's important to make people aware of the fact that as technology advances, it's not just helping us keep in contact or get from point A to point B quicker than we could do before. It's actually helping us understand what it is that defines us as free-thinking, conscious individuals.
Right, okay, so that just gives you an example of a monogenic syndrome of, that causes an increase in appetite, which then leads to an increase in eating and body weight. And what we've been able to do is look at these spontaneous sort of syndromes where in mice and different species, and to try and understand what factors are impacted that then leads to changes in appetite and body weight. And so thus far, when we look at factors that are influencing obesity, as I've said already, we've got this balance, we've got changes in food intake, we've got energy expenditure, which is comprised of physical activity and thermogenesis and basal metabolism. And then we also have energy storage, so fat absorption and fat metabolism. But most of the, thus far, the obesity medications have focused on this energy in part of the equation, looking at how we can reduce appetite. But new medications on the horizon are looking at ways of increasing thermogenesis to try and burn calories without physical activity, without exercise. And one of the only current obesity medication in the UK at the moment is the lipase inhibitor. So what that's doing is it's preventing dietary fat from being absorbed when it's taken in through food. But what I'm going to talk to you today is about the energy and the appetite component of this circuit. And the first thing I'm going to talk about is leptin. I'm also going to talk about serotonin and pro-opioid melanocortin. But let's just start with leptin here. And what is leptin? Leptin is a hormone that's produced in body fat or adipose tissue. And the more body fat we have, the more leptin is produced. So when we have higher levels of leptin, that then signals to the brain, and we get then a consequent reduction in food intake. And that's this kind of cycle that occurs in normal weight, healthy individuals. So this was really exciting when leptin was discovered. And the way that it was discovered was looking at this, as I was mentioning, these kind of spontaneous mutations. So way back in 1949, there was a really, really chubby mouse, which was named the obese mouse, or OB because it's missing. So, you know, it's obese is what it was called for the 2 copies of the gene. But it wasn't known what it was. And these mice were 3 times the weight of their siblings, and they ate twice as much food. And then in 1994, Jeff Friedman cloned the leptin gene and named it leptin, and realized that it was produced by adipocytes. And then in 1997, the first humans were identified that have leptin deficiency, which means, they can't make leptin, or they have virtually non-existent levels of leptin, and they have profound obesity. And taking that same child and providing it with leptin replacement therapy then restored appetite and body weight. And this was really, really exciting at the time. And people, in the obesity field were thinking, wow, okay, we've just figured it out. We've found something that is so essential for appetite and body weight regulation, and we can use this to treat obesity. The next thing that was discovered was the receptor that it acts through. So again, there was a mouse and also a rat that had this spontaneous mutation, and this mouse was named the diabetes mouse, or DBDB. And likewise, it was 3 times the weight of its of control mice. And these mice, like the OBOBs, they also suffer from severe type 2 diabetes. The leptin receptor was cloned in 1996. Humans with leptin receptor variation were identified in 1997, and humans with the full leptin receptor mutation were identified in 1998. And both leptin and leptin deficient humans are incredibly rare. But this allowed us to find a really instrumental factor in regulating appetite and body weight. Unfortunately, it hasn't turned out to be a great treatment for obesity. And that's because, as I said, with extra adiposity, there are higher leptin levels. So obese individuals already have higher leptin levels. So giving obese individuals leptin as a treatment, although it was tried, wasn't as effective. And that's because the individuals developed something that we think of as being, leptin resistance. And so the current target is looking at ways to improve leptin sensitivity or reducing leptin resistance. So to just summarize, leptin is a hormone that's produced by adipocytes. It reduces food intake in general. If mice or humans don't make leptin, it causes profound hunger and hyperphagia and obesity. Leptin replacement therapy can ameliorate this. However, it hasn't been a useful treatment for common obesity because of elevated leptin levels. Leptin signals through its receptor, and genetic mutations in the leptin receptor also cause profound hyperphagia and obesity. And it's actually the version of the leptin receptor that's found in the brain that is involved in the regulation of energy homeostasis. And so leptin does more than just one thing. One of the things it does is regulating appetite and body weight. And it's via action in the brain where this occurs. So can we harness the inverse relationship between leptin and food intake? It hasn't worked out as well as we'd hoped. So current research attempts are looking to develop drugs to increase leptin sensitivity. Okay, so let's move on to the next thing then. Another neurotransmitter and pathway that has been really instrumental in regulating appetite is the neuropeptide, or the prohormone pro-opiomelanocortin, or POMC. And what POMC does is it's produced in a variety of different tissues, but it's the one in the brain that we're most interested in. And in particular, it's expressed in the arcuate nucleus of the hypothalamus, and it's also expressed in the nucleus of the solitary tract. And we know that this is really important for regulating appetite and body weight, because mice and humans that don't make POMC are obese and hyperphagic. And in fact, a recent report also indicated that many chocolate labs are deficient in POMC as well. And you might, if you have ever known a lab, then you'll know that these dogs can be very, very hungry. So POMC competes with, or POMC peptides, because as I said, it's a prohormone. POMC peptides compete with agouti-related protein, which is also found in the arcuate nucleus of the hypothalamus. And this is a really essential hunger hormone or hunger neuropeptide. And these two compete for action at the melanocortin 3 and 4 receptor. And so you have increases in POMC reduce hunger, whereas increases in AGRP increase hunger, and the two compete for action at the melanocortin 3 and 4 receptor, with the melanocortin 4 receptor being the one that's the most dominant. So what does the arcuate nucleus of the hypothalamus look like? Well, it's at the base of the brain, and you can the two types of cells, they sit they're sort of neighboring, they sit near each other, and they talk to each other, and they release their peptide products at primarily the melanocortin 4 receptor to impact appetite to maintain energy homeostasis. And they're getting signals from the stomach, they're getting signals from the GI tract, they're getting signals from the pancreas, they're getting signals from the adipose tissue. So this is one of the ways that leptin, which we just talked about, is acting to affect food intake. So I mentioned that that one of the ways that we found out about POMC is looking at mice and humans and dogs that don't make POMC. And these individuals are develop obesity at a very young age. And one of the ways that we know that the signaling is through the melanocortin 4 receptor is, again, because people and animals that are deficient in the melanocortin 4 receptor are hyperphagic and obese. And this is what the mice look like. And these are males and females, with the knockouts being the top black lines, the heterozygous being in orange, and the wild types in gray at the bottom. And you can see here that this disruption in melanocortin 4 receptor signaling causes a significant increase in food intake. And so this was first reported in 1997 in mice, and it was followed up in humans in 2003. The humans with melanocortin 4 deficiency were identified. So there were two groups that found these, that identified these humans at the same time. And what we now know is that monogenic, the disruptions of the melanocortin 4 receptor are the commonest known monogenic causes of human obesity, representing approximately 5 to 6% of severely obese people. So whereas leptin deficiency, leptin receptor deficiency, POMC deficiency are all very rare, melanocortin 4 receptor deficiency is a little more common, but still, 95% of people who are obese won't have disruptions of the melanocortin 4 receptor. But what we've been able to do is take advantage of this knowledge that the melanocortin 4 receptor is really important in regulating appetite and body weight, and develop medications increasing the activity of the melanocortin 4 receptor. And so this is a new drug called setmelanotide that's been developed by Rhythm Pharmaceuticals, and it's currently being investigated for the treatment of rare genetic forms of obesity. So a little bit of a success story there. So whereas leptin didn't work out, hasn't worked out yet, I should say. I mean, still people are very much pursuing this as a possibility. The melanocortin 4 receptor already is being tested in humans. So moving on to the next aspect, which is I'm going to talk about serotonin, or 5-HT. And one of the reasons why I'm interested in 5-HT is because it's been known for a long time to impact appetite and body weight. And serotonin was first discovered back in 1948, and it was named serotonin because it was found in the serum and it caused constriction. And what shortly thereafter, what was discovered is that there's an inverse relationship between serotonin and food intake, in particular brain serotonin and food intake. So one of the things that I've been asking over my career is, can we harness this relationship between serotonin and food intake to improve obesity? So some of my first research that I ever did in my life was looking at whether or not a medication like Prozac, which increases serotonin levels, whether or not it can be used to reduce food intake and body weight. And in rats, it does reduce food intake and body weight. And it was a similar idea to a drug that was actually licensed, which was called Redux, which people were very excited about at the time because what it did is it increased the bioavailability of serotonin. So by blocking the reuptake of serotonin and stimulating the release of serotonin, more serotonin was available to act at its receptors. Unfortunately, it was removed from the clinic, and that's because of unwanted side effects. So by just globally increasing serotonin everywhere in obese individuals, there was an increase. There were some individuals who developed valvular heart disease, and so the drug was withdrawn. So one of the questions that I asked early in my career is, how is it working? So which receptor is it acting through? So can you dissociate the therapeutic benefit from these unwanted side effects by, instead of just globally increasing serotonin, more specifically acting at a particular receptor? And serotonin receptors have been clustered into 7 different families based on sequence homology and effector pathways. And just from pharmacology studies, we knew that the serotonin 2 receptor family was really important in regulating food intake. And the first knockout mice that were created that didn't make the serotonin 2C receptor developed this hyperphagia and obesity phenotype. So it told us that serotonin has to act at this serotonin 2C receptor. Otherwise, if it can't, then you get an increase in food intake and body weight. So one of the things that we can do now is we can look in more detail at how and where serotonin might be acting to affect food intake and body weight through the serotonin 2C receptor. And we can use this new technology, which is called chemogenetics. And what happens is in chemogenetics, and this particular type is called DREAD, or designer receptors exclusively activated by designer drugs. And in this, in DREAD, what we have is we have our normal, so we've got our serotonin acting at its serotonin receptor, so that's fine. But in chemogenetics, what happens is with protein engineering, you end up with this designer receptor, and that designer receptor no longer binds the regular neurotransmitter, but instead binds a designer drug with exquisite precision, and that allows you to control the activity of different subsets of cells. And so what you do is you inject your AAV DREAD into a particular brain region or a particular tissue. I'm just giving an example here of TPH2 eye cream mice, for example. And what then happens is that you get the receptor, which could be a GQ receptor, it could be a GI receptor, whatever kind of receptor you want. That binds then to those specific types of cells. And then you can administer your designer drug whenever you want, and you can turn the cells on and off as you like. And so we use this technique to try and identify the serotonin 2C receptors, specific subpopulations of these receptors that were important in regulating this effect that we saw with the knockouts. And we found that a key population of these serotonin 2C receptors is on the POMC cells, which I just told you about before. So you can see that through all of these different techniques, what we're finding out is that there's, there are these key circuits. So just to say, so which serotonin receptor, we know it's the serotonin 2C receptor that's really important. And then we're looking at regulating the activity of which cells. And as I've just told you, I just gave you a little preview here that it's the melanocortin cells, it's these POMC cells. So this is the model that we came up with is, so how is the drug that was just withdrawn, from clinical use, difenfluramine or Redux, how does it work? So we're thinking that it stimulates the release of serotonin, acting at serotonin 2C receptors in the arcuate nucleus, impacting the activity of these POMC cells, which then can signal at the melanocortin 4 receptors. And so we were able to demonstrate this a long time ago in 2002, that this was how difenfluramine worked. And this, but as I said, what I've been interested in trying to understand is, well, how does difenfluramine work? And one of the things that researchers have realized is that it's through the serotonin 2C receptors, and then this led to a serotonin 2C receptor agonist being approved for as an obesity medication. And unfortunately, it's now been withdrawn in the United States as of February this year, but it's still telling us this important circuit. So how is it improving obesity? Is it working the same way as difenfluramine? Is it also acting through these POMC neurons? And so again, this would be the model, is that lorcaserin, this obesity medication, would be acting at the serotonin 2C receptors, influencing the activity of POMC and allowing it to signal at the melanocortin 4 receptors. So to test whether or not this is how it works, what we did is we took some mice that either had functional or non-functional POMC. So you can see here that the POMC neomice don't make POMC, and we crossed them with these serotonin 2C receptor cream mice to allow us to have mice with fully functioning POMC, mice without POMC, and mice with POMC restored just in the subset of cells that express the serotonin 2C receptor. And in this case, we see lorcaserin reduces food intake, as you would expect in wild-type mice. It reduces food intake in control Cree mice. It does not reduce food intake in POMC, in mice that don't make POMC in the arcuate nucleus. And this effect is restored if we restore POMC only in the subset of cells that express the 2C receptor. So that tells us how the therapeutic benefit of lorcaserin is being produced. We then looked at the next target of POMC, which would be the melanocortin 4 receptor, and again, used genetic models to test this. So we've got wild-type mice and we've got mice that don't make the melanocortin 4 receptor. If we give lorcaserin to wild-type mice, as you would expect, you see this reduction in food intake. And if mice don't have a functioning melanocortin 4 receptor, then the drug doesn't work anymore. So this tells us that the way that lorcaserin is acting is via these POMC neurons signaling to the melanocortin 4 receptor. So what we've been able to do is we've been able to kind of decode how these two obesity medications that were in the clinic, diphenfluramine and lorcaserin, act to produce their therapeutic benefit. They act by signaling at POMC neurons, which then signal at the melanocortin 4 receptor. So whether it's diphenfluramine that's stimulating the release of endogenous serotonin, or lorcaserin going in and acting at the 2C receptor on its own, the end result is that they're both acting through the melanocortin circuit to reduce food intake. So that takes me to the summary slide. So in summary, as I mentioned at the beginning, overweight and obesity are now the norm in many countries, such as the UK and the US. And this is having a detrimental effect on health, and it's shortening lifespan by approximately 9 years. It's increasing susceptibility to disease. So what we want to know is we want to try and understand, how is appetite regulated? How is body weight regulated? Now, most people can improve their health with just diet and exercise alone. So just being more aware of how much food their bodies require, and making sure that they try and adjust how much food they're eating to just what their bodies require. But for some people, this is incredibly difficult for a variety of different reasons. And so other treatment options are really important to help people promote good health and reduce disease and treat disease. So when we think about how to affect appetite, you might first immediately imagine thinking about targeting the gut. But in fact, what ends up happening is all of these signals, whether it's the nutrients themselves, the hormones that are produced by fat, the hormones that are produced by the gut, hormones that are produced by the pancreas, all of these different signals are coming into the brain where they're being integrated. And what we've been able to do over the past 20, 30 years, 40 years, is identify key brain regions, and then within those key brain regions, key circuits that are impacting food intake, appetite, hunger, body weight. And then by identifying those key circuits and pathways, what we can then try and do is develop medications to take advantage of them, to harness their power, to be able to correct food intake and then cause a reduction in body weight. So this has then led to medications, this new knowledge that has been arising over the past, has arisen over the past couple decades, has allowed us to develop medications that are now in human use. So I just think it's really important that we continue to try and improve upon this and continue to try and identify these key circuits and novel ways of tackling and harnessing these circuits within the brain so that we can come up with better medications with fewer side effects. So of course, I'm giving you not only some examples of my own research, but also a lot of other excellent scientists over the decades. And so it's been a real privilege to be in this field and to know some of these people as well. So I just want to thank you very much for your attention, and I would love to take some questions.
Excellent. Thanks so much, Laura, for the fantastic presentation. All right, so first question here. Christiane has asked, lorcaserin was recently removed from the market due to an increase in cancer. And so, Laura, do you know what the relationship is between 5-HT and cancer, if there is any? And do you think it would be possible to bypass this adverse effect somehow?
Right. Well, in fact, it is a little bit difficult to understand why the FDA chose to issue a warning about lorcaserin. Actually, the drug company voluntarily withdrew the medication. The difference in the incidence of cancer wasn't statistically significant between the placebo treated group and the lorcaserin group. So lorcaserin doesn't statistically increase the risk of cancer. So yeah, it hasn't been demonstrated, and it hasn't been reported. It's very hard to understand why the FDA issued the warning, because the difference wasn't there. So yeah. There isn't a clear link between serotonin and cancer. Yeah, but you can also just think about, their medications targeting the serotonergic system are very widely used globally.
Interesting, thanks. Here's another question from Francoise. They've asked, is there a future for leptin LAPR pathway regulation for combating obesity? What do you think about that?
Yeah, it's definitely a very active area of research. So what people are looking to do is they're looking to increase the sensitivity of the leptin receptor. So it's such a cool idea. If you can imagine that in obesity, leptin levels are elevated. So people already have the leptin on board to produce a beneficial effect. The idea would be to impact the receptor to increase the sensitivity of leptin acting at its receptor. So that's where the drug discovery efforts are at the moment. And I think, yeah, it's a fantastic idea. So watch this space. It could happen. Another way that people are addressing it is to try and understand specifically which subset of leptin receptors are involved in regulating appetite. And that study, some of that research has been pioneered by Martin Myers at Michigan, and he's looked at leptin receptor in a variety of different locations in the brain. And what we've, in reading his research, what's become evident is that there isn't one subset of leptin receptors that's really the most important. They're all kind of working in concert together to affect appetite. So it's unlikely that there will be like a specific brain targeted region of leptin receptor, but more likely it will be just like more globally increasing leptin receptor sensitivity. But, we're still, his lab and a lot of other labs, Brad Lowell's lab, are still looking to try and figure out whether or not there is a particular subset of leptin receptors that are really, really important, and then you can go after those.
All right, great answer. Here's an interesting question. So you talked a bit about some genes that affect obesity. Do you have any experience looking at epigenetic factors, how environmental factors can affect epigenetics?
That is a whole other area of research. So yeah, I mean, people are certainly very interested in that and thinking about, individual, within a single individual, how these epigenetic factors are impacting food intake and, appetite, obesity, but also, maternal influences, paternal influences, multi-generational influences. So yeah, it's a very, very active area of research as well, thinking about what might be happening in that. So yeah, that would be a whole nother talk.
Yeah, I imagine. Good question here from Karen. She's asked, how do SSRIs, so I think selective serotonin reuptake inhibitors, how do they affect appetite and weight?
So SSRIs, that was one of the slides that I showed you. So that's what I started with, as I started with SSRIs. If you give SSRIs to, if you give Prozac to a normal weight animal, what you'll see is a reduction in food intake and a reduction in body weight. What happens with people who are taking SSRIs for other, for another condition? Well, in that situation, then you can get anything. You can have an increase in food intake and body weight. You can have a decrease in food intake and body weight. You can have no change. And that's because people who are taking SSRIs, if you think about it, if they're working, if those medications are working and improving whatever condition the person might be taking them for, whether it's anxiety or depression or something like that, the reason why the medications might be working is because there's a dysregulation in the serotonin system. And so the medication will be correcting that dysregulation. So if you increase serotonin levels on a baseline, from a particular baseline in a your average rat or your average mouse then what you would find is an increase in serotonin levels and a decrease in food intake. But if you're starting at a different baseline, what you might be doing is just increasing them back up to normal. But also, if you have conditions like depression, some people with depression eat more, some people with depression eat less, some people with depression don't change their food intake. So you don't get a consistent effect in different patient groups with SSRIs.
Interesting. All right. Perfect. Here's a question, and it might be related. So stress and anxiety.
I should.
Sorry, go ahead, Laura.
I was just going to say that SSRIs in obese people reduce food intake and body weight. That's what difenfluramine. That's what difenfluramine did. So in an obese individual, SSRIs will reduce food intake and body weight. But if you're taking it for something else, you might not see that.
Okay, perfect. And I think in the interest of time, we'll make this next question the last one. But Anna has asked, stress and anxiety, for example, can affect food and weight intake. And so how would you expect for stress to play a role in these pathways?
Yeah, that's a great question. In stress, some people eat more, some people will eat less. It depends on the particular stressor. It depends on the person. It depends on the situation. But in general, if you're thinking about fight or flight, if it's a very, very stressful situation, then appetite's turned off, because we need to not think about, oh, I'm really hungry. We need to worry about like getting away from danger as quickly as possible. So it depends on the kind of level of stress you're thinking about. And if it's, just general daily stress or, general environmental stress, then it can have a different impact depending upon how people choose to cope with that stress, with some people possibly eating more and other people eating less, and the people eating more, maybe eating more comfort foods, which are higher calorie foods. So you kind of have to, in studying this and thinking about it, it's important to kind of drill it down to answering a very refined question, a very specific question. But these brain regions that are involved in fear and anxiety and stress are also involved in appetite as well. But, because as I said, like if you're in a situation where you're trying to get away from danger, then you want to have your appetite turned off. Your hunger
Great answer.
turned off.
All right, fantastic. Well, thanks so much, Laura, for the fantastic insights today, both during your great presentation as well as the Q&A session.
I just want to thank everybody for coming along to the webinar, and it was just a real pleasure. And so thank you to everybody for taking time out, and I really enjoyed sharing this area of research that I really love with all of you.